probably in the introduction, I probably say something that he did in the beginning. I just, this is the work I did during my postdoc at Columbia University in the Ivailo Ivanov lab. So we try to understand the specific role of innate lymphoid cells. We know that these cells, as José told us, is very difficult to study because they are very cool because they have a lot of functions, but they are very difficult to study because they don't have like specific markers. So we try to understand these cells in this context. So basically, here's as José told us, the ILC3 mirrors the CD4 T cells. We have different flavors of innate lymphoid cells that share the transcription factors with T helper cells. So as you can appreciate here, we have innate lymphoid cells type 1. They express Tbet as Th1. They lack TCR and BCR specific B and T cell receptors, but they share the transcription factors with the helper cells. So we have ILC2s that are innate lymphoid cells that share the transcription factor GATA3 with Th2. They are very important for the helminths and allergy reactions. And the focus here, we would like to understand how the innate lymphoid cells type 3 works. So basically they have like, let's say a master transcription factor RO gamma T. They share with Th17 cells. They can produce IL-17 and IL-22. They are very important for the bacterial response, autoimmunity, and during fungal responses too. So we know that all of these subsets, we have like, José told us, different subtypes of innate lymphoid cells, all depend of IL-7 cytokine during development and to keeping these cells proliferating and developing. They express all of the RO gamma transcription factor, and some, for example, this can be distinguished by the NCR positive ones. They express NKp46, and the other ones that are called NCR negative or CCR positive that express the chemokine receptor. We have one special one that now is kind of classifying another type. It's called lympho-tissue inducers. They express RO gamma 2 as the other ones, and they can produce the cytokines. They can produce lymphotoxin alpha and beta. They are very important for the lymphoid and pyrespec development. And basically all these subtypes, they produce cytokines IL-22 and IL-17. Here's just like we have different strategies to study cells. Basically, they are lineage negative. So we have to stand for lymphoid lineage, dendritic cells, macrophages, and B cells. And you kind of get in the lineage negative. And after we can analyze the IL-7 receptor, the CD9.2 expression. So basically here we have all the IL-6 gate. So we can distinguish the three types based on the transcription factors that they express. We have Tbet1 is the ILC1s expressed. We have the ILC2s that express GATA3, and you have the innate lymphoid cells 3 that express RO gamma, and some express Tbet2. So here we can define these three populations that José just mentioned to us: the NKp46 positive one or NCR positive, the CCR6 one positive, and this one that is we call double negative ones. So these cells are very important for the Barreian function, and they reside in the lymph nodes too, and the Pires patch, but mainly in the guts. So we have here, they can reside in the Pires patch, they can live in the lymphoid tissues like mesenteric lymph nodes, but they kind of locate mainly in the lamina propria. So we have in the large intestine these structures that is called cryptopatches that they are important too. So basically they are in the interface with the interface in the gut. They are important not just for the pathogens, but to keep the commensals controlled too. So this just a summary of all the functions that have right now we kind of know about the innate lymphoid cells. We have they are important in the responding to metabolites and the circadian rhythm. They can produce IL-22, and they are these metabolites and circadian rhythm, they can control the development and the roaming of these cells. We can, as we saw early in this Jose presentation, they are important for the neutrophils, and they can produce IL-22, IL-17 that are important in the barrier function and the production of antimicrobial peptides by the intestinal epithelial cells, the proliferation of these cells in tight junctions, and the recruitment of neutrophils. But they are important too in the production of IL-2 that are important for the keeping or maintaining regulatory T cells in the gut, and they are important in the production of GM-CSF2. So the main cavity for our field is kind of difficult, as Jose show us before. We don't have like many models that we can delete specifically the innate lymphoid cells. We kind of have to use like depletion antibodies or using like other markers that could delete some subtypes, but we don't have like a whole mouse that is ILC3 depletion. So basically one of the classical models we use like a depletion antibody against Ty 1.2, and we use RAG as a recipient mice, and we deplete these ILC3s and we transfer the T cells and B cells for these mice, what is congenital mark like Ty 1.1, and you deplete these ILC3s, and you have like T cells and B cells, but you don't have ILC3. Main of the papers say that, but basically they don't have the other ILCs too, right? Because they express, as I show you guys before, the Ty 1.2 marker. So we don't have like so many specific models. And so we trying this project to address this very difficult question, but what are the specific functions of ILC3 in the gut immunity in this context? How ILC3 function in the presence of T cells, and are the T cell responses similar in the absence of innate lymphoid cells, type 3 innate lymphoid cells? To do that, we have to generate a mouse model that we call here origami stop mice. We invert the origami gene expression and lock this gene with two LOXP sites. One is a common one, and one is a like called stop one. So we call these mice origami stop mice, and we cross these mice with CD4 Cre. So doing that, we turn on the origami expression in T cells. So basically, this gene is inverting the correct orientation, and the origami could be expressed in the cells that express CD4 Cre, but they still turn off in the origami innate lymphoid cells type 3. So we start to first want to understand how is the T cell development in these mice, because we know the origami is very important for T cells too. So as you can appreciate here, when we analyze the stop mice, basically they behave like origami knockout mice, a full knockout mice. They have like thymomas, they die after like 16 weeks, and they have defects in the CD4 T cells development. So basically, as you can appreciate here, they lost the CD4 positive single positive cells. They have a defect in the double positive in the CD8 maturation. And then when we cross the mice with CD4 turning on the origami expression in innate lymphoids in CD4 T cells, we can rescue these population. So we have here normal numbers of CD4 single positive cells, the CD8 maturation, the double positive numbers. So next we want to understand especially in the small intestine, the intestine. So this is the small intestine analysis. So we get in the CD4 and TCR beta positive cells. As you can appreciate here, we have RO gamma expression and FOXP3. And in the wild type, we have all the three populations that express RO gamma and FOXP3. So we have the single one, the double positive, and the FOXP3 regulatory T cells. In the stop mice, we lost all the T cells that express RO gamma in the small intestine. And basically in the RO gamma stop mice, or CD4 mice, we rescue this RO gamma expression in T cells, as you can appreciate here. And you have normal numbers of regulatory T cells comparable with the wild type. So next, the most important, we want to know if this mice is really ILC3 depleted. So we call these mice delta ILC3 after we confirm that. So as you can appreciate here, we have gated in the lineage negative gate, as I mentioned before, the strategy I showed you guys before. We have here GARA3 and RO gamma expression. So they didn't have ILC3s and the frequency and the numbers, showing that these mice are really ILC3 depleted. So the conclusions we have in this part, we observe that the T cell development is recovering delta ILC3 mice. Th17 cells differentiation is restored in the delta ILC3 mice. ILC3s are absent in delta ILC3 mice, but the most difficult part, as I told you guys before, this when you deplete RO gamma, you lost the lymphoid tissue inducers, the cells. So these mice don't have lymph nodes. They don't have lymph nodes as a wild type because they lost all the ILC3s, even the lymphoid tissue inducers. But we want to understand in this, let's say in this condition, how this happens. So later I will tell how we kind of overcome this difficulty. So the next step, we want to understand the profile of these cells, especially the T cells and the ILC3s in these mice. As you can appreciate here, we have we did a bulk RNA seq in the CD4 purified CD4 T cells from the gut of these mice. So we have here the mice that are delta ILC3. They look very similar to the wild type, the CD4 T cells. As you can see here, we recover all the transcripts from the CD4 T cells. They express IL-17, IL-22, CCR6, the IL-23 receptor, and the RO gamma expression. But when we did analyze the intestinal epithelial cells, we did scrape, we isolate these cells and did a bulk RNA-seq. True, and we can appreciate here that most of the antimicrobial peptides, they are depleted in these mice or are very low expressed, showing that ILC3 basically controls the epithelial genes or the antimicrobial peptide production, as is expected by the literature data. So basically, the conclusions we have here, the ILC3 control genes are down in the stop and the delta ILC3. IL-22 response genes are down-regulated the ICs of stop and delta ILC3 mice. And the Th17 cells program is restored in the CD4 T cells from the RO gamma delta ILC3 mice. So next, we want to understand in the infection context how the T cells and the ILC3's contribution are different or are similar what we know in the literature. So we use the Citrobacter urination model. There's a classical model to study ILC3s. As you can appreciate, here's what we know in the literature. So ILC3s are well known to be important in the beginning of the infection. They produce a lot of IL-22 in the beginning. But over the infection, the pathogen kind of grows up the levels of the pathogen, and the T cells took over and usually produce IL-22 and kind of clear this response, this infection after 2 or 3, usually 3 weeks. So we want to understand the mice, the IL-3 role in these conditions. So when we infect with the normal or regular high dose of the Citrobacter rhodesianum, all the mice die. They ate. They have higher numbers of CFUs in the feces and the tissues, and but we try to use a low dose to see if we can solve some differences in especially in the mice that have T cells. When we did that, we can observe, you can appreciate here, we have like 3 days, they can survive longer, the delta LS3 mice compared to the stop mice. So we want to understand these differences in vivo. But the problem here, as I mentioned before, the wild type mice survive, but basically these 2 mice don't have lymph nodes, and the wild type have lymph nodes. So we want to put the mice in the same condition. So we did a bone marrow chimera using as a recipient Arrow-gamma knockout mice, and we sublethal delete these bone marrows, and we transfer bone marrows from the wild type stop and delta LS3 mice. So the main goal here is all the mice don't have lymph nodes and have different cell types. And we did infect these mice with Citrobacter rhodesianum. As you can appreciate here, we can still have the same difference between the delta LS3 mice compared to the stop mice. But as we expected, the wild type mice die too without the lymph nodes, showing that as we expect, right? So lymph nodes are very important and crucial for this clearance, because this pathogen needs to T cells to clear this pathogen. So to show that this difference are due to the CD4 T cells, we did the same thing, and we depleted using a depleting antibody for the CD4 T cells. We deplete these cells in the delta LS3 mice. As you can appreciate here, we lost these differences when we deplete the CD4 T cells. And one interesting thing we observed too, we work with a commensal called SFB, segmentofilamentous bacteria. So in our mouse, we have all the mice are SFB negative, but we have some mice that are SFB positive. So we try to understand if the T cells from commensal are important in this context too. So we infect these mice that has SFB or not. So as you can appreciate here, we saw that some of these differences due to in the presence of CD4 T cells that stimulated by the SFB. So to confirm that is IL-22 dependent, this effect, we did the same experiments, and the Arrow-gamma stop in the delta LS3 mice. As you can see here, when we deplete, using a depleting antibody for the IL-22 cytokine, we lost the differences between the stop and the delta LS3 mice. And when we did try in this, the context of the delta in the bone marrow chimeras that I mentioned before, the Arrow-gamma knockout mice, we saw the same differences when we treat the mice without the treatment with now is with the IL-22 cytokine. When we did treat the mice with IL-22, we can observe here the stop mice, they survive longer, and the wild type and the delta LS3 mice are very similar, showing that IL-22 is really important for this infection. So in this part, we conclude that IL-3 are required for the protection at high dose. Both IL-3 and T cells contribute to early stage for low dose infection, and the lymph nodes are required for the clearance of Citrobacter rhodesianum infection. So next, we try to generate a model that we could have, still don't have IL-3, but we have lymph nodes and Pires patch. So to perform that, I did create a mouse crossing aerogama flox mice with a ubiquitous Cree that express ubiquitous Cree that express the Cree-ER that is tamoxifen inducible. So we treat these mice after they generate the lymph nodes with tamoxifen, and when they get old, we analyze they lost a lot of innate lymphoid cells and T cells because of the deletion of aerogama. But to confirm with this depletion, we did a bone marrow chimera. We had these mice and did transfer the wild type, stopping the delta LC3 bone marrows. So in this context, we have all the mice have lymph nodes and Pires patch, but they have different contribution of T cells and ILC3s, as you can see here. So basically, the delta LC3 mice just don't have delta LC3, ILC3, sorry. So we did confirm first that these mice have normal Th17 cells development. So they have, as you can appreciate here, they have Th17 cells and FOXP3 regulatory T cells. They lack LC3s in the guts. So this is the small intestine and the large intestines too. So as you can see here, the numbers are really down. They don't have. So we would like to understand now we really have a mice with lymph nodes and especially we don't have ILC3s in these mice. So basically we did start with high dose first to see what happened. So when we infect the mice with Citrobacter rhodesiae at the high dose, they die at early stages as we observed before. Basically, it's due to the presence of IL-22 from the aerogama's ILC3s that express aerogama. And but we want to understand the same condition during the low dose because we saw some differences in the early stages that maybe T cells are important too. So we did infect the mice with the low dose. When we did that, the mice survive. As you can appreciate here, we have the stop mice, they die earlier, but the delta LC3 mice with lymph nodes and Pires patch, they survive. They did very bad, and when you look like the weights, and we have like inflammatory cells like neutrophils and macrophages, they have more, higher numbers and inflammation in the gut, but they survive after the low dose infection. As you can appreciate here, we did analyze the T cells in the early stage for T cells in the later stages. You can see here we have lower production of IL-22 and especially in the T cells from the large intestine. But when we did analyze later in the large intestine, we can observe there is no differences in the IL-22 production and the interferon gamma 2. So to try to understand these differences, we want to understand the context of a commensal host interaction. So we took advantage in the lab. We have a mice that is specific for SFB that is called 7B8. It's an oncogenic marker. We could sort these cells and they are GFP, IL-17 GFP. So we did transfer these cells for the different recipients. And these mice are already colonized with SFB. So when we did analyze Th17 cells in this context, we can observe there is not significantly differences in the mice that don't have ILS3s. So the conclusions overall we have is we generate a ILC3 specific deletion in mice. ILC3s are required for protection of Citrobacter rhodesiae infection, the higher burden. But Th17 cells can provide protection for Citrobacter rhodesiae at a low dose. And in using these mice, we can study the relative contribution of ILS3 and T cells in the mucosal immunity. And so this is what we did. I want to use these mice, but I have to come back to Brazil. But one important thing we want to understand, especially in the context of like mucosal or vaccine immunization, how is I think is one of the main questions in the literature about the ILC3 contribution. We know that we have like trained ILC3s that are already demonstrated, but we don't know in the context as like a ILC3s like antigen presenting cells in the T cell context. How is the impact of like in the, like say in a vaccination or a challenge response? So I'd like to thanks all the people in the Ivanov lab, the lab I did my postdoc in, the Ivail Ivanov, for the opportunity to do the postdoc there. And our collaborators, Harris Wang and Yimi Huang, that did help us with the commensal analysis and the RNA-seq analysis too. So I'd like to thank you for your attention.
Thank you, Leandro, for this great talk. It was I found very, very fascinating this genetic tools for depleting ILC3. So I was wondering, you mentioned at the end of your talk, so this is all in inflammation, but and did you check T-Rex? Because we know that some Bro gamma T ILC3s, others peptides or Janus cells can promote tolerance. I'm just curious if
Yeah,
in
I
that, like
checked.
in that animal, like without
We
inflammation, did you see anything related?
Yeah, we did check the regulatory. So I didn't check, let's say, in the context like tolerogenic or like say, let's say a diet or something that we could understand. Looks like they are kind of involved. They have a little. I probably saw in the fax plot, right? They look a little smaller, the population. I think for sure they are important. So one thing I didn't do, but we want to do in this project, we want to see these, especially the ones that are APC ones, right? They express MHC class 2, but we didn't have the time is the same. Sometimes we have to just publish, right? So we are close
Yeah.
to kind of finish the project, and I was coming back to Brazil, so I didn't have too much time to kind of test this. But this is one thing I really want to do, is like, especially in the tolerogenic context and the re-challenge, right? So we didn't test these mice. I don't know right now if somebody in the lab is doing something related to that. I think maybe not, but this one thing I would like to do there, but I didn't have time.
Can I
Yeah. Yeah.
Oh, sorry.
Go.
I want to follow up in a question following yours. We know we now kind of we have a new cell type now that is basically dendritic cells that express Rho gamma T.
Yeah.
And I was wondering, I don't know, it's a lot of things for checking. You did a wonderful work, but by any chance, did you check this RORC disease, like this Rho gamma T positive disease?
Yeah, we did like what we did at the time. Just did like a population. I put this. I didn't. So I didn't put here because it's a very short presentation. When we did analyze, like let's say populations numbers, like we didn't see any differences. But for sure, will be helpful if you saw like probably they will have some impact, right? I feel.
Yeah.
I think
Yeah,
in
because
the model that we try, the Citrobacter, maybe then for sure the dendritic cells are really important. So we could even have more, like let's say differences or something, but it's kind of tricky to do the ILC3 like
yeah,
model, right? Because they share so many things like with like
yeah,
disease or T cells, and we didn't know about disease right now. It's like a new thing, right? So it's kind of
yeah, like 2 years ago, 1 year ago.
yeah,
Yeah, immunology is always like that. Like whenever you
yeah,
think you're close to something, there's a new paper showing that what you think is true, it is not true. So I'm just asking because we kind of we so you can clearly show in your paper that you can deplete specifically LC3s. You don't have like a very much effect on TA17. But consider that this dendritic cells that also express Rho gamma T, and it's a
yeah,
very little population, but apparently they are very important. They are playing some roles, mainly by
yeah.
by priming like Tregs. So that's why I was wondering if you check, because could explain what Eugenio just said. Like like, did you check Tregs? If you have a change in Treg, does it really mean that is ILC3s? Could be this raw CDCs that is doing something. I know
Yeah.
immunology, we cannot never exclude one variation. You always have like the other things on the background interfering
Yeah,
what you have.
that's the trick thing, because some people, we know dendritic cells, but they know, like, let's say NKT cells express RO gamma 2 right now. Mate cells express, right? We have a lot of other gamma delta cells express RO gamma 2, so it's kind of very tricky to work with ILC3. So I think maybe right now the model that you used, like the delta, the HR, NKP40, some people are using this Cree as a, like, but you deplete just one of the populations, right? So the question we want to ask is, like, let's say the whole, or the whole LC3's population, how this happens. For sure, yeah, I think this is one thing that we could follow up. We didn't do that there, but I think there is a lot of things we can do right now. If I feel that in this, especially this, the APCs ones, right? The ones that express arogamma, and they're important for comment, so they can delete T cells. I show here, like, in the commensal context, but for sure in the diet, or, like, let's say tolerogenic models, they will have some difference for sure.
Thank you, Leandro, and thank you, Jose. I don't know if there's any more questions. Thank you so much, Jose and Leandro, for this great talk. And very, I wish you the very of the success in all of your career, and hopefully, we'll see you in person.
Jose will be here, right, Jose? Soon.
I
Yes.
hope.
I hope
Yeah,
you
I hope. I hope.
Hopefully.
Yeah, but thank you a lot for the invitation. It was very nice. Like, as I told you in the early beginning today, it's very important for us, first authors, sharing our work to make our names kind of famous in the world, because we're just starting. So we want to get like acknowledged for what we're doing. So I really appreciate this opportunity for sharing the whole work behind the first author doing.
Yeah, that's a very good initiative, right? So it's very nice, and I'd like to thank you, and I'm very happy too, because we can discuss science, and it's a very important thing, right? Too, and we can kind of know each other. I know Jose just for the papers, right? Because I was
Yeah.
in US, I didn't.
Yeah. I know you because of the position. I saw your name, and then I kind of crossed your name with the paper that came out last year, because I don't know, our paper came out at the same time. So basically